Friends of the Dar, ladies and gentlemen, good evening. We are delighted to welcome you to Dar al-Athar al-Islamiyyah. And tonight we are privileged by the presence of Dr. Bahareh Azizi, who you may be familiar with as she has been leading as a volunteer the science programs for children for 3 years now. Tonight she will be talking about science, and specifically where science and art combine. Her title will be Beauty Lies Within, the Art of Protein Structure. Dr. Azizi is currently a consultant at the Kuwait Foundation for the Advancement of Sciences, and prior to joining KFAS, she was the head of basic science research at the Dasman Diabetes Institute here in Kuwait. Dr. Bahareh Azizi is a research scientist and fellow member of Georgia Institute of Technology in the United States. Let me remind you to turn off your mobile phones, please, and join me in welcoming Dr. Bahareh Azizi to the podium.
Thank you, Ivina, for that kind introduction, and thank you all for being here today. I know that this lecture is a little bit outside the typical themes that you host here at the Dar al-Athar, but my many thanks to Sheikh Hassa and the Dar al-Athar team for giving me the opportunity to present my version of art, which is looking at protein structures. So I don't think I have to do much convincing that we all know that nature is indeed beautiful. And, oops, excuse me. Let me just press escape. Okay. And most of you have marveled at the many things that nature has to offer. Of course, the different animal species there are beautiful flower species. My husband, who loves the outdoors more than I think he loves being with me, has many pictures of which he just adores nature, and in a sense, I admire it through his eyes as well. I think Vincent van Gogh said it nicely. He said, "Keep your love of nature, for that is the way to understand art more and more." But what I'm going to be talking to you about is nature that's not necessarily seen with the naked eye. This is nature that's beautiful on a very, very small level. We call it the molecular level. And I want to tell you a bit of story about how I came to love protein structures. It was my first year of graduate school at Georgia Tech, and I was taking a class called macromolecular structure. I had no idea what macromolecular meant at that time. I hate to admit it. It really just means proteins. But I thought to myself when I first signed up for the class that, oh my God, I am going to have to sit dreadfully through this class in which I'm going to learn about some sort of structure, and really how beautiful can structure really be. And so Professor Hudd, who was my instructor at that time, started the class by putting up this picture. And the first thing I thought to myself was, oh my God, that is a beautiful piece of art. And he then went on to explain that this is one of the most beautiful proteins that has been discovered to date, and as we go through this course, you're going to learn more about what each of these little things mean and how it all comes together, and really, proteins are a form of art. And it was that statement and perhaps that picture that really changed my life, because in a sense, at that time, I wasn't really sure what I wanted to study. I didn't know if I wanted to get my PhD in metabolism or cancer therapy, but it was this picture that said, I want to study protein structure, and I want to find a way to discover more and more of these structures. Now, please keep that in mind, because in a few slides, I'll tell you how that all changed. So proteins, most of you have proteins as a part of your regular diet. Most of you have probably seen on the side of cartons that give you nutritional facts about the content of protein within that specific item that you're eating. But on a smaller scale, if there's any work that needs to be done in a cell, most of the time that work is done by a protein. And you probably have come across different types of proteins because you've heard of hormones. Many of you have heard of the hormone insulin. Diabetes is a big problem in Kuwait, as you know, and you probably know the people that take insulin shots. Hormones are a protein. There are proteins that are called enzymes. These are proteins that just take something and make it into something else. There are these proteins in our cells that are called receptors. There are antennae. They feel what's going on inside your body, and they adjust things in your body accordingly. And of course, when you get sick, you know that your body has antibodies, and those fight off basically the virus or the bacteria that's in your system. So the antibodies are also a form of protein. And you have these channels, which I'll show you in the next slide, that basically transport things in and outside your cells. And of course, muscles, hair, skin, your eyes, all of those are comprised of proteins. And the only reason why I'm telling you that is because there are so many different types of proteins. And when you start to look at the structure, which we will in a few minutes, you'll see how each of these structures are so beautifully designed so that they're able to do their function. And that is a key thing to keep in mind. So we talked about membrane channels. This is an example of a membrane channel. If this is a cell, this is the inside, this is the outside, this is a protein. And we'll take a look at a few of these structures in a bit. But proteins are found in all living things and even in viruses. So a lot of the time when you get a cold, what infects you is the virus's protein. So there are proteins in their structures as well, and some of them have been solved, and I'll show you one of HIV towards the latter part of my talk. And we talked about antibodies, which are shown as these Y-shaped things here. This is a virus in blue. When you get sick, your antibody will attach to the virus, and eventually your immune system will clear it out. Now, the difficulty in proteins, as I said earlier, is that they're extremely small. So if we just take a look at this scale here, here's the blue whale, the largest mammal known. Here's a human. And if we look at an ant, which is about 1 centimeter on this scale, proteins are about 10 million times smaller than an ant, which is hard to comprehend because when you look at science, it's really hard to appreciate what happens at that small scale because you really can't see it. And so for a long time, it was a challenge to figure out what these actually look like because technology had to become advanced enough so to allow us to see what these proteins actually were structured. And most of you, or perhaps some of you, have seen the famous Watson and Crick structure of DNA, which was solved in 1953. At the same time, the scientist Linus Pauling from Caltech solved the first component of proteins, which is called the alpha helix, and I'll show you what that looks like in a second. Now, both of these groups used a technique called X-ray diffraction. And what that is basically you take X-rays, you shoot it at this molecular structure, whether it's a protein or a DNA, and from that you can decipher what the structure actually looks like. Now the issue is, because proteins are so small, shooting just an X-ray at one protein molecule doesn't work. So what we have to do as scientists is we have to form these crystals. We take these proteins and we make these crystals of these proteins. Why? Because in a crystal, you not only have one protein molecule, you have an array of the same protein molecule repeated. So when you basically shoot X-rays at them, you can get a clear diffraction pattern, which I'll show you what it looks like in the next slide, that'll allow you to determine what the structure is. Now, just as a quick note, forming crystals of proteins, extremely challenging, takes a lot of time. A lot of my friends who were getting their PhDs in protein crystallography probably spent a good 1.5 years praying over the protein trays that these crystals would form. Extremely, extremely difficult. So if you just take the crystal of the protein, what you do next is take it to a cyclotron. This entire building is where the protein gets shot with all these different X-rays. So you need an infrastructure that big. It is really an amazing building in itself, and of course the technology is quite advanced. And what you get is this pattern that looks like that. Now, I told you a few slides ago that I wanted to become a protein structural chemist. I wanted to discover the different structures of proteins. And then I took a class on this, how to determine the protein structure from a diffraction pattern, and I nearly failed that class. So I realized, yep, not for me, definitely not discovering protein structures for me, but I can focus my PhD on using protein structures that have been determined to find ways or new drugs to bind these proteins. So this changed my life in a different way as well. So back to this, from the diffraction pattern, you get some, I guess scientists basically what they do is they can determine the structure of these proteins by forming these electron density maps. it's not really important, but eventually you get your model that looks like that. So I just want to show you how complicated these diffraction patterns are. If this is the molecule that you're trying to discover the structure of, when you shoot it with the X-rays, this is what the pattern looks like. So scientists actually take this, break it down, and then determine that that looks like a duck. It was very hard for me to do. I really didn't do well in that class. And I probably shouldn't say that in front of my mother, but it's true. Another example is a cat. If you shoot at X-rays at a cat molecule, this is what the X-ray pattern would look like. So I say this so we can appreciate all the efforts that actually go into looking at and discovering these molecules. And the last point I really want to make is because we're able to discover what these proteins looks like, it has really changed our understanding of modern medicine and human health. And so why this is important, imagine there is this illness that's due to a protein not functioning properly. In the past, before you had these structures, what you would do is perhaps hypothesize how that protein works and then make a drug and see if it worked or not. Now what you can do is you can visualize, you can actually see where that drug would bind to the protein, and you can enhance it by making those interactions stronger so that the drug can be more effective. And that's really changed the way that we look at disease and understand disease as well. Now, most of you have heard of the Human Genome Project, that the human genome has been sequenced. This is only going to advance and revolutionize this process even more. Before I show you some structures, I have to pay respect to the very fundamentals of proteins themselves. So just to give you a brief background on the basics, there are 5 atoms that make up most proteins: carbon, hydrogen, nitrogen, oxygen, and sulfur. The main point is that when you combine these, phosphorus here is found in DNA, but not in proteins, you form the basic building block of proteins called amino acids. Now, many of you perhaps take supplements, protein shakes that have amino acids in there. This is because you need amino acids to build strength, build your muscles and tissues, and so on. But really what it is, a protein is a chain of different amino acids. And there are 20 amino acids that make up the 19,000 to 20,000 proteins that we have in our body. Now, just for a second, if you can do some math, you can realize that there are only 20 that make such a diverse group of proteins that really allow our bodies and living things to function the way they do. So that's pretty cool, if I should say so myself. So these 20 amino acids form these things called these amino acid chain that eventually becomes the protein. Now, the order in which the amino acids come is very important. It's like your name. If you were to change the order of the letters in your name, it would no longer be your name. And it's the exact same thing with proteins. If you change the sequence of a protein, the sequence of which the amino acids come, you can get a regular protein and you can change it and it can actually become a diseased protein. So if you've heard of sickle cell anemia, which is very common, if you look at our regular blood cells, there are 7 amino acids in a row. If we just look at the 6th amino acid, in the normal red blood cells, it's glutamic acid, one of the amino acids. With sickle cell, what happens is this glutamic acid, which is normal, changes to another amino acid called valine. And just changing that one amino acid takes the normal red blood cells from their circular shape, and it makes them sickled. They become longer, hence sickle cell anemia. So the sequence is extremely important, and that's something that we have to keep in mind because every protein has its own unique sequence. Now, in a minute, I'm going to show you some of my favorite protein structures, and hopefully you can appreciate some of the art encompassed in them. But in order to do that, you're going to see some different shapes going around, so I just want to identify those shapes to you. Proteins are made up of 2 main shapes and 2 minor shapes, as we like to call it. They are the alpha helices, shown here as a ribbon. Okay, so just remember ribbons. And they are these arrows or beta sheets or strands. So in the structures that you'll see, it'll be a combination of both. It'll be predominantly only one. It'll be predominantly both, one or the other. And what you'll also see is these things called loops and turns. And loops or turns are just connective. So from one ribbon to the next, you have what's called basically a turn, or sometimes you'll have a loop, which is basically almost a full U. So we've gone over the primary chain of amino acids, and then what happens, as I just mentioned, they form either a ribbon or the arrows, and then they all come together and they form what's called a tertiary structure. So they just come and form their little, the structure itself. And then there's some proteins that have the same tertiary structure repeated, and they form what's called a quaternary structure, which I'll show you, it'll make more sense. Hemoglobin is a good example of that in a second. Now, proteins just don't come together. There is no way for us to look at a sequence of amino acids and know what it's going to look like. What we can do is look at the sequence of amino acids and make some hypotheses based on what types of amino acids are there. But there are some scientific principles that allow proteins to fold or come together. And I just wanted to show you a few of them, but the main one I want you to keep in mind is that our bodies are made predominantly of water. We're about 70% water. Our cells, in which the proteins are mainly found in, are about 70% water as well. And that's a very important point, because the first thing that determines how a protein actually folds or comes together is whether they like water or not. So some amino acids are very hydrophobic. They do not like water at all. And some amino acids are very hydrophilic. They love being around water. So what does that mean? If you have a chain of amino acids, when it comes to fold, remember they're going to be surrounded by water. So naturally, all the amino acids that don't like water will come into the center. They will close themselves away from the water, and they'll be surrounded or protected by the amino acids that do like water. So that's one example of what determines how proteins fold. The hydrophobic, the ones that hate water, are mainly found inside of the protein, deep inside. And those that enjoy water are found on the outside. The second thing is interactions. We all remember this from school. Positive attracts negative. And too much positive and negative repel each other. The other thing that proteins or atoms in general are very finicky about is personal space, similar to us humans. Someone comes too close to you, you want to push them away. Atoms are pretty much the same. So there's a space that you have to keep, or else they're going to clash with you. So interactions, very important. That positive-negative affinity, extremely, extremely powerful when it comes to forming protein structures. The second one, or a different type of interaction, is the hydrogen bond. So most of you know James Bond, haha, this is a joke. Hydrogen bond is when a hydrogen atom interacts with an oxygen atom. That's also a very powerful bond. And the third one is called the van der Waals interaction. Really, this is just to say that sometimes atoms just like each other temporarily, and it's really a temporary effect, and they can move away from each other and so on. So that also determines whether they come together or not. Geometry is very important. This just tells you that amino acids can rotate to a certain angle. Sometimes they can rotate a little bit more depending on the nature of the atom, and sometimes a little bit less, but geometry is important. And last but not least, very, very importantly, that proteins, when they fold, they will take the path of least resistance and lowest energy. So when it comes to protein structures, we want the lowest form of energy. Right? We don't want to put a lot of work to form a structure. It has to, in a sense, and I don't like to use the word natural, but it has to be natural in the sense that it doesn't require a lot of energy when it actually finally forms. And so that's it. We just took our primary sequence. We learned about the two main structures, the ribbon and the arrows. They form these tertiary structures, and eventually the quaternary structures, which I'll show you about in a few minutes. Now, I decided to take a few of these structures that I've always genuinely liked, Some of them have great function in terms of the immune system or in relation to cardiovascular disease, and some I just think are just really, really pretty. So the first one is a protein from barley seeds. I'm going to give you the scientific aspect of the protein, of course, but I also want you to appreciate that it looks like, in this case, as a star. So if you look at this protein, it's actually not that big, and you can see that it has the same repeating unit 6 times, so it's what we call a hexamer. It has 1 ribbon, and it has a few of those arrows, and of course, it has the loops that go around. This protein is an enzyme, as we talked a little bit earlier about. They take something and convert it into another. A lot of what happens in this protein happens within the center. That's that open space. So I like this one because it looks like a star. The Moco protein shown here resembles somewhat of a star, but this is what is basically a trimer. And this enzyme is involved in metabolism. So you'll see it a lot in relation to the metabolic pathway. As a matter of fact, if this enzyme is actually mutated, it can lead to a neurological disease that can be quite deadly. So this small protein can have an adverse effect if the protein is not structured properly. Now, I like lectin because it looks a little different. It's predominantly, actually almost all of those arrows rather than ribbons. You don't see very many ribbons in lectin here. Lectin is a protein that's basically used for cells to talk to each other. That's why you can see in this little picture here, the blue lines are lectin. It allows for cell communication. It also allows sometimes to get toxins, because actually some form of lectin can actually be quite toxic, and some are actually good for you. So you have to be careful when you're eating them as well. Now, my favorite protein, I would say, of all of them is this one right here, which is ApoA1. Now, don't have to worry about the name very much, but most of you know of HDL and LDL when you go to the doctor and they give you your cholesterol readings and the good cholesterol. Well, ApoA1 is part of the good cholesterol. This is the good cholesterol, basically, HDL, and that's ApoA1 shown here in orange. The beauty of this structure, if you look at it, it's so long in terms of its ribbons. I mean, it goes for a really long time. If you look at the other pictures that I was showing earlier, the ribbons were actually quite small. But this is a really long and elongated alpha helix or ribbon, and it reminds me of a horseshoe or the infinity sign, which I thought was pretty cool. Cystatin C, which is basically a biomarker for kidney function. So a lot of the times when you go to the doctor and they assess your kidney function, they're looking for the presence of this protein. I think this protein is really interesting because it looks like a pair of wings. And for me, when I look at this protein, it's actually one of the ones in which I see motion, whether it's the way that the arrows are formed or whether the way that the structure comes together. To me, it's quite enticing. But it does have a very important role in kidney disease and more recently found in cardiovascular disease as well. Now, hopefully you don't know anyone that's ever had pneumonia, but if you do, this is the cause of pneumonia, this protein right here. So people that actually get sick from pneumonia are because this protein has infected their cells. Now, it's not very nice because we don't want to think of pneumonia, but the protein itself is quite interesting. It has these 2 long ribbons that stick out, and it's surrounded by a few ribbons and some arrows. And I think it actually looks like a butterfly, if you can imagine these being the antenna of the butterfly and these being kind of the wings. So this is also why it's one of my other favorite proteins. Now, in a few minutes, I'm going to switch to an online version of this where I can show you the protein actually moving. Hopefully the internet will work. But this is a propeller fold of a protein, and it resembles a propeller of a plane. And in this case, there are 7 different units you can see colored. Some of these propeller proteins only have 3, some have 8. But they're really beautiful in terms of the way they actually function, because in a sense, let me just go back, in a protein like this one, there are different units that have come together With the propeller proteins, this is one long amino acid chain. So it's one unit that forms this propeller shape, which I find quite fascinating. Now, I think this is one of my other favorite protein designs, as you can see, primarily because of the intricacies of all the details of the structure. Now, this is a complex of different proteins that have come together. This protein specifically is very much involved in immunity. And you can see that there are so many dimensions to it. You have structures that are protruding out. There is a propeller shape inside here, basically the same fold. Same fold that I showed you. I'm still going back. Same fold that I showed you earlier. And you can also appreciate that within the pocket itself, or inside the circle, there are also some ribbons floating around. So very, very detailed and quite complex, because the immune system, as you can imagine, is also quite complex. Insulin, again, we talked about insulin at the very beginning. Most of you have probably heard about it. This is what it looks like. Now, this is an insulin hexamer, which means there are 6 insulin molecules within this structure. A single insulin structure looks like that. So basically 2 ribbons and an arrow. What you see in the center here is that the insulin molecule protein is actually binding zinc. And this is how it's believed insulin looks like inside our pancreas. So this is kind of what we would say are the native form. Now, if you know anyone that has had cancer, for example, a lot of the times you'll probably hear of this term called p53 tumor suppressors. These are proteins that basically stop cancer cells from dividing. And how they do that, now the protein is this ribbonly structure shown here. This structure in the center is DNA. Because the way this protein works is that it binds to the DNA and does not allow the DNA to then make more and more cells. So it's a tumor suppressor in that sense. And I find this to be really beautiful because it shows basically the connection between the protein and the DNA, and a function that really, in a sense, saves our lives. So it's extremely important. Now, one thing I want to remind you, perhaps I discussed a little bit earlier, was about the positive-negative affinity for each other. Well, DNA is negatively charged. So all the protein residues, or all the protein amino acids that are touching the DNA, are mainly positively charged. That negative-positive interaction is what keeps these structures together. The vault is a very complex protein structure. I was trying my best to find a way to show you smaller pieces of it because it's quite intricate and quite detailed. But then I thought I'd show the whole thing because it's really magnificent when you look at it. It looks like 2 ski hats coming together in my perception. But all of these intricacies are all these different protein structures coming together to basically form this magnificent protein. And this protein is also involved in immunity. Now, alpha hemolysin is a protein that basically causes sepsis. And the interesting thing about this protein is, if you look at it from the side, it looks like a palm tree. It's got the base here, and it's got the branches, in my opinion. But fascinatingly, when you are infected by staphylococcus, which basically causes sepsis, what happens is this protein will come, and it doesn't have the top part yet. It has only the base, and it'll come and it'll attach to your cells. And it'll go inside the membrane of the cells. If you recall, I told you that cells have a membrane. And it'll form somewhat of a hole. And then it'll form this branch, that part that you see up here. But if you look down the hole, it looks like this. This is the top view of that actual protein. So because it causes a hole, what ends up happening is all the contents in your cells start to leak out, which is what causes you to have sepsis. And in some cases, pneumonia as well. So a beautiful structure can cause great damage. And speaking of membrane proteins, I don't know if many of you know this, but our membranes are extremely fatty. They're made up of lipids. And so what happens is when you have a membrane protein that binds to the membrane, if this is very fatty, which means it doesn't really like water very much, you can imagine any protein structure that's inside the membrane is also going to be very hydrophobic. They also they won't like water. They want to be around, they want to be protected away from the water. However, the proteins that are outside of the membrane into the cells, now they're surrounded by water, so the majority of these proteins are hydrophilic. So again, you can make some deductions by understanding these concepts as to how these proteins look like. And the same goes for channels, which we talked about at the very beginning, that transport ions into and out of cells. The same thing, you can see here that the majority of the part of the protein that's inside the membrane is very fatty, very hydrophobic, and the part that's outside is very much exposed to water. Now, I like Betabarrel proteins. They are also found in the membrane. I like them because they're predominantly made of the arrows that are anti-parallel. They form a barrel shape. It's exactly what it looks like. They're very commonly found in transporting of items into and out of the cells. Everyone knows of the HIV virus. This HIV trimer is what actually causes the virus from being so deadly, in a sense. So I wanted to show this, of course, for the disease context, but also, despite the fact that it has a behavior that's not so much, in a sense, liked, it is an interesting protein to study. And many scientists have tried their hardest, actually, to figure out what the structure of the HIV proteins are like, because only then can they discover drugs that can overcome it. And the last couple that I will show, this is the BRCA1 protein is a protein that's involved in breast cancer. A lot of people who test for breast cancer susceptibility, you will test to see whether you have this protein. And this is what the protein looks like, and it's very important because if the protein is mutated, and this is in combination with another protein, you can see if you just take a look over here, if this is the normal protein, once it becomes cancerous, you notice that there is a change in the way the structure is down here versus the normal. And this is often what causes it to become leads to cancer. My research was done on these proteins called nuclear receptors. I'll come to the previous slide in a second. I love these receptors. I think they are, in a sense, probably the prettiest proteins, but they're not the prettiest proteins to actually show an audience who hasn't really seen protein structure. But I love these proteins for one main reason, and that is, first of all, they're involved in everything. They're very important in disease. If you've heard of the estrogen receptor, they're implicated in cancer. Another receptor is implicated in diabetes. But I want you to appreciate that these proteins actually undergo a conformational change. Okay? And what I mean by that is the way these proteins work is they bind small molecules. So the estrogen receptor will bind estrogen. Without the presence of estrogen, the protein looks like this. Just pay attention to this purple ribbon. So once the protein actually binds estrogen, or the molecule, which is shown here in blue, what happens to the purple ribbon is that it shifts. It will actually come into the pocket and close it to protect the ligand so it doesn't actually leave the protein. So this conformational shift then allows for the other processes in the cell to occur. And if that shift doesn't happen, then the rest of transcription or DNA or the pathways for moving forward don't also occur. And so sometimes when these proteins are not functional, this shift doesn't happen. So a very, very small change can make an enormous difference. And the last protein I'd like to show is hemoglobin, but I think it's probably best if I show you hemoglobin. Let's see if we have the structure. Hopefully it's still working. Yes, so this is the hemoglobin protein. This is a program that we scientists use to basically look at the structures because we can go deep inside the pockets and move it around and play around with it. I don't want to give anyone motion sickness, but what you can see here is hemoglobin you're all aware of. Hemoglobin basically transports oxygen. Hemoglobin has 4 heme or basically these heme molecules have an iron center, which is how hemoglobin works. So you can see that in the hemoglobin molecule, there are 4 hemes shown here. The structure is predominantly ribbons, right? And there are 4 different units that come together. So you can actually take a look at just the single unit itself. So that's one. And the last protein I'd like to show you in full motion is this one. As I was trying to decipher which of the proteins I'd show you, I thought this one was actually quite cool. It's a little bit different. And we can shift it to look at it from a side angle. This little molecule here is the ligand for the protein, or basically something that binds it. And it allows us to appreciate the fact that simplicity sometimes is key as well. Some of these other proteins I'd shown you were so intricate, but there are proteins that are quite simple, quite small, but have a very important function. So I hope that through this presentation, I've been able to kind of shed some light into a different form of art, one that has existed in nature for a very long time, that we've only had the benefit and privilege of actually seeing within the past 60 to 70 years, but that really it's the very small, very, very, very small things that are really beautiful and that can make a really big impact into how we function. And so I, again, thank you for the opportunity, for allowing me to share my passion with you. Thank you.